Hi everybody, this is Miss Dewey. Today we are going to talk about cell division, specifically the cell cycle, mitosis, and meiosis. But before we get into the details of these particular processes, I want to talk about why we even care. Why do cells divide in the first place? So there are three main reasons that a cell needs to divide. The first is growth. You started as a baby, but now you are much bigger. That is because your cells divided and helped you grow. You also need your cells to divide so that they can replace lost or dead cells. So whenever you get a cut, you're actually losing some cells, and cell division is how we replace those cells. The last reason is to reproduce. Now, when it comes to reproduction, different organisms do it in different ways and use different cell division processes. So there are two big types of cell division. The first is mitosis. This is what some organisms use for asexual reproduction. We don't do that, but some do. And this creates identical copies of cells. So we use mitosis for other processes in our body, but not for reproduction, because we use meiosis for sexual reproduction. Meiosis makes unique cells, which is important to give our offspring more differences, to make them genetically different, but we will talk a little bit more about the details of that process in a moment. For now, we want to talk about what happens for cells to divide in the first place. Cell division is a complicated process, so cells need to get ready to divide, just like you need to get ready for a big test. So in order to divide, cells have to go through the cell cycle. And there are three big things that happen in the cell cycle. In the cell cycle, the cell will grow, it will duplicate its DNA, and it checks for mistakes. Each of these things happens in specific stages of the cell cycle. So there are three phases that we consider interphase in the cell cycle. We have phase G1, gap 1. This is where a majority of the growth happens. We have S phase, which stands for synthesis. Specifically, the synthesis of DNA happens here. So S phase is where we make new copies of DNA. Finally, we have G2 phase, gap 2. The cell finishes up its last bit of growth. In each of these steps, there is also a checkpoint. So the cell will actually stop the cell cycle, check to make sure that everything is okay, and then keep going if everything is going as planned. If the cell doesn't do this, then this can actually lead to issues like cancer because you'll have cells that have mutations, and that can be very bad. Our final phase of the cell cycle is M phase. This stands for mitosis or meiosis, depending on which type of division you're doing. So in mitosis and meiosis, the cell and the nucleus divide, but cytokinesis is that final step where the cells actually finally split off and become two totally new cells. Right now, I want to just really quickly zoom in on what happens to the cell during S phase. So this is DNA synthesis, when the DNA replicates. It does this through base pairing. So what will happen is you'll have one strand of DNA, the strand will split apart, and you've got your base pairs on each side. You've got your A, T, C, and G. And then what'll happen is other nucleotides will come in matching that A, matching that C, matching that T, and it will make two totally new strands. We consider DNA synthesis semi-conservative, semi meaning half, and conservative meaning old, because of each strand that you have, half of that strand will be old and half of it will be new. But why do we need to replicate DNA in the first place? Why is this such an important step? So every cell in your body needs its own set of DNA. Your DNA controls what your body is able to do. It controls what the cells make. So you have to have that set of DNA in every cell in your body. So if we're going to make two new cells, then we need two sets of DNA. So what you see on the screen right now is what the chromosomes look like before S phase. So you can see that it has one strand, and I tend to say it looks kind of like the letter I here. Then after S phase, they look something like this. They've got two separate strands that are attached at the middle, and it looks a little bit like an X. So each of those two strands are identical. We've just copied the DNA over. Because they are still attached, we consider it one chromosome. Each of those strands we consider to be one sister chromatid. So when we talk about splitting up sister chromatids, we're saying that we're splitting up these two strands. So now that we know what happens before mitosis and meiosis can happen, let's start to look in at a little more detail at these processes. So mitosis is how new identical cells are made. This is happening in your body all the time because it's how your body grows, it's how it repairs, it's how it replaces cells. And not in us, but again, in certain organisms, it can be used for asexual reproduction. So the process of mitosis, there are 4 major stages. There's prophase, metaphase, anaphase, and telophase. If you need to remember this, you can remember it by the acronym PMAT. So in prophase, we have the cell, and it starts out looking like cells normally do, where you've got the cell and then you've got the nucleus. But in prophase, the nucleus will start to disappear. This is so that the chromosomes can come out and be separated the way that they're going to be in mitosis. Then in metaphase, they start to line up in the middle of the cell. I always remember metaphase because that's when the cells line up in the middle. They both start with an M. Next, we go to anaphase. So that's when the sister chromatids start to be split apart, when we have the 2 strands of each chromosome split apart to become their own chromosome. Finally, we have telophase. Those chromosomes are on each side of the cell, and the nucleus starts to reform around each set. Then, after mitosis is done, we have one final stage called cytokinesis. So when we say cytokinesis, this is when the cell is cut into 2. So that is mitosis. Again, it ends with 2 identical cells. Next, let's talk about meiosis. So it's really important that you understand the purpose of meiosis. Meiosis is the process that makes gametes, egg cells and sperm cells. And gametes are special because they are haploid. They have half the number of chromosomes that our normal cells do. Our normal cells are diploid because they have 2 sets of chromosomes, but gametes only have 1 set. This is because when you have an egg and a sperm come together, you only want half the DNA so that when they come together, they make something that has 1 full set of DNA, or 2 sets total. So meiosis is special because it actually divides the number of chromosomes in half. It ends with the haploid cell. The way that meiosis creates haploid cells is by doing 2 divisions rather than 1. So meiosis will actually create 4 cells at the end, which have half a set of DNA. The other thing that is special about meiosis is that all of the cells that are made at the end are different from one another. This is why if you have siblings, you probably don't look identical to those siblings unless you're identical twins. It's because the cells that are made from meiosis that are used in reproduction end up with different bits of DNA in them, so that when they come together during sexual reproduction, you end up with offspring that look different. So again, there are 2 divisions in meiosis. There's meiosis 1 and meiosis 2. Each step does PMAT, prophase, metaphase, anaphase, and telophase again. So if you see prophase 1, prophase 2, that's definitely meiosis because each step happens twice. So just to review, cell division is important for 3 big reasons. It is important for growth, it is important for replacing cells, and it is important for reproduction. 3 things need to happen before a cell can divide. The cell needs to grow, it needs to copy its DNA, and it needs to check to make sure that there are no mistakes. There are 2 types of cell division. In mitosis, we're making identical cells. Mitosis is used for cell growth, repair, and it's also used for asexual reproduction. We also have meiosis, which is used to make the ingredients for sexual reproduction because the cells that it makes are haploid and genetically different. That's all that we have to say about cell division. Again, I know I went through this pretty fast. This is a lot of information. So if you have questions, you can either reach out to me, you can look at the slides that have been linked in the description, or you can look on Schoology where we have some more information that you can look through to learn about this topic. I hope that everybody is doing well and that you have a great day.